So today we're going to be talking about, I guess we're not talking about bacteria and archaea yet. So we briefly touched upon this last class when I showed you guys the different classifications of life and talked about phylums. So we're going to be reviewing that. So even though we are different from like rats, we're both considered mammals, and mammals are a part of class Mammalia. So this like distinction between us and rats, but also this like mutual mammal characteristic is a result of classifying life. So how we classify life is determined within this study called taxonomy. And taxonomy is based on grouping life together based on their shared characteristics. So again, going back to us and rats, because I really like rats for some reason, we are both mammals. So that means that when we give birth, we make milk to feed our kids. And that's like a shared characteristic, even though we're not rats. Sorry, I've said rats so many times in the past like minute. Anyways, so there are 7 main classifications of life from broadest to the most specific. And so the modern father of this classification is a scientist called Carl Linnaeus, and he classified a lot of organisms and discovered a lot. And these 7 classifications from broadest to the most narrow are domain, kingdom, phylum, class, order, family, genus, and species. And then so on the right, you see an example of the different classifications and how they work out. I just realized Zoom has like an annotate tool, so I'm going to be using that. How do I use it? Spotlight? Is that how I use it? Can you guys see my cursor? What is happening? Oh my god, wait, what's happening? Oh, whoa. Wait, sorry, I'm like Zoom grandmaing right now. So, oh my god, wow. Wait, what am I? Okay, never mind. We're not going to be annotating because I have no idea what is happening. Anyway, moving on to the next slide. So the broadest classification of life is something called a... Sorry, I'm still trying to get this. Okay. Can you guys see like the red thing moving around the screen?
Yes.
Okay. Okay, that's good. Okay, so there are 3 domains. Oh my god, I accidentally clicked. Okay, so this is the broadest classification of life. So this means that every living thing can be classified into either eukarya, bacteria, or archaea. These are the 3 domains. And so the main differences between these few domains is something called organelles. So organelles are membrane-bound structures in our cells. So all life is made out of things called cells, which are like building blocks. And then within these cells, we have like different little compartments. So we can think of the cell as like a factory and organelles as different machines. So eukaryotes have these machines that do specialized functions, which allows them to grow with more complexity because they have all of these different parts working for them. They can focus on doing other things like growing. But hang on, let me just admit some people. So, okay. So we have many different examples of organelles, like the nucleus, which is where all of your genetic information is, which gives rise to your traits. And then you have your mitochondria, which is the powerhouse of the cell. So eukaryotes or domain eukarya, they have these organelles. but then prokaryotic domains. So the remaining 2 domains are prokaryotic. So that means they don't have these organelles, but it's not just eukarya and prokarya. You have eukarya, and then you have bacteria and archaea. Bacteria are bacteria, and then archaea are like bacteria, but they're kind of different. They used to be classified as bacteria, but then scientists realized that there were a lot of differences. If you see the diagram on the right, this is some examples. This is an example of some of the organisms from these 3 domains. So you have eukaryota, or eukarya, archaea, bacteria. So if we look at the anatomy of a prokaryote, so anatomy is how it's like our body parts and our bodily functions and how all of that jazz works. So a prokaryote is obviously very simple. We have all of these skeletons, organs, and different organ systems, but all they have is one cell. So another thing is that prokaryotes are always unicellular. Okay, so someone just asked, I guess it's not a question, but someone just said, I don't understand what archaea are. So for now, just think of them as like bacteria that are not like really bacteria that we know less about. So like lesser known bacteria. We'll definitely go into it as we go on with the slides because that's the focus of today's class. Yes, so they are extremophiles, as Vedant said. So basically just think of them as rarer bacteria that live in like more extreme environments. So like really salty or really hot or really cold environments. So you have this prokaryote over here, and basically the few things that you need to remember are, first, we have the cytoplasm, which is where everything is suspended. Then we have the plasma membrane, which is like, if the cytoplasm was like water, the plasma membrane would be like a cup. Okay, flagella are really important because they're like, they help bacteria move because they don't have legs, but they still need to get around somehow. So they have this flagella thing. And then, yeah, that's pretty much all you need to be aware of. So this is a really, really pixelated comparison chart of eukarya, eubacteria, and archaea. So eu just means true. So true bacteria versus archaea, which are like fake bacteria. And so these are some comparisons. Does anyone have any questions? Because we probably don't know what any of this means, and that's okay, but not going to go too deep into it because don't really need to know what this is. But if you have any questions, I'd be happy to clarify. Just gonna pause here for like 30 seconds. Okay, moving on from this very beautiful graphic. So I was really out of brain cells when I made this PowerPoint, and that's why your lovely title over here is Archaea. Anyway, so what are archaea? So archaea are unicellular prokaryotes. That means they don't have organelles that do specific things for them, and they're only made up of one cell. Again, they used to be really confused with bacteria because they kind of look the same, as you can see from the right picture, but then they found different cellular, genetic, really small leveled differences, and that's why they differentiated archaea and bacteria. So it's an example of this would be, so plants have cell walls that protect them and make them really rigid so they don't tilt over. In the same way, bacteria have cell walls that also help them maintain their structure and protect them. And these cell walls are made out of a special compound called peptidoglycan. Peptido meaning protein, glycan meaning sugar. And then Archaea don't have these peptidoglycan cell walls. Instead, they are the only source of biological methane, which is a gas found in the atmosphere. So Illinois asked, was that a typo or simply doing the Archaea thing? Yeah, that was on purpose. I'm sorry, I am really the walking definition of cringe. Anyway, so the main unique thing about Archaea is that they live in super, super extreme environments. So like super hot, freezing, acidic, basic, salty, deep in the ocean, gamma, UV radiation, you name it, they probably live there. No one else probably lives there. And that's why it's really hard to study them. Because I personally would not like to go 200 miles deep into the ocean crust to see some bacteria looking things. But yeah, it's really hard to study them. And that's why we don't know much about them. So compared to the bacteria slides, these are going to be a lot shorter. So we have a couple major groups of Archaea. First, we have the extremophiles, which are basically all bacteria. But we don't really have a name for them like we do the other groups. And also another thing to note is that I showed you the 7 Carl Linnaeus classification sections, but really like bacteria and Archaea don't really follow that. Instead, they're just split into groups called clades because, I mean, we don't really know much about them. So the few different types of extremophiles include, but are not limited to, halophiles, which love salt. So the prefix F or PHIL means loving. So yeah, extreme thermophiles, which as you probably guessed, means that they love hot environments. And then methanogens means that they like methane. So when we inhale oxygen and then we go through cellular respiration, which gives us energy, we release carbon dioxide as a byproduct. But then methanogens, or it might be methanogens, I have no idea. Anyway, they're poisoned by oxygen. So instead, they release methane and they live in swamps or marshes where okay, it's hard to explain this, but basically you have a swamp, and then on top of the swamp you have like a bunch of plants, and then these plants like release all of the oxygen and stop oxygen from reaching under the swamp. And so methanogens who are poisoned by oxygen will live in these swamps and then release methane. Whoa, sorry, almost fell here. Okay, anyway. Yeah, and that's what marsh gas is. And so these are very informal classifications, but if you want to go more scientific-y, you have clade, Archaeota. Some thermophiles also belong in that group, but they mostly belong in crane Archaeota. Archaeota are the oldest living organisms. Nanoarchaeota are organisms with the smallest number of genes ever. I cannot pronounce that. So, yeah. Okay. Done with Archaea. So basically, in a nutshell, Archaea are bacteria that we do not understand. Okay, so what do bacteria look like? So we looked at the general structure of a prokaryote, and they're generally all going to follow that, but they have a bunch of special features that we have not talked about yet. So first, their size, they are 0.5 to 5 micrometers in diameter. On the other hand, our cells are about 10 to 100 micrometers. But the thing is, like, we are made out of trillions of cells. they're just one cell. So you can imagine how small most of them are. They have a cell wall made out of peptidoglycan. Again, it's a protein combined with sugar, and it's unique to bacteria. So when we get bacterial infections, it's really easy to target the peptidoglycan in bacteria without killing our own cells. And so antibiotics are designed to stop peptidoglycan from forming in the bacterial cell wall. And remember that the cell wall is like a protective layer for the bacteria. So when the cell wall disintegrates, the bacteria are more prone to being attacked by white blood cells and other immune defense systems. So other than the cell wall, you also have these things called capsules. So capsules are a sticky layer of carbs or protein, and it is dense and well defined. So that means that it's like really, really compact. It's not like loose. Yeah. But if it is loose, then it's called a slime layer. And these capsules, which are these sticky layers, they help different bacteria come together, forming like a colony or a protective surface. So an example of this would be, you know how on your teeth you sometimes get like white plaque? That's called a biofilm, and that's a bunch of bacteria sticking to each other. So these colonies also protect against loss of water or dehydration, shield pathogens from attack, because I guess it's harder to beat like 30 than it is to beat 1. Fimbriae, don't know how to pronounce that. Anyway, so those are hair-like appendages with sticky uses. So this just means that they help bacteria stick to surfaces. And so there's also something called a pili or pili. Pili are used for like transferring genes from one another, which we'll look at in another slide. And then flagella, again, helps with movement, and they evolved from pili. So the genetic information stored in bacteria has been a very interesting research topic in the past few years, because although they have less DNA than eukaryotes because they are more complex, they have these circular things called plasmids. And plasmids are these self-replicating DNA molecules that carry a few genes. So it's separate from the entire genome of the bacteria. So the region of the cytoplasm occurs or appears lighter than the nucleoid, which is nucleoid, and within the nucleoid, the actual genes are contained. And these genes are contained in the forms of DNA and proteins called chromosomes. So yeah. Okay, reproduction and adaptation. So bacteria and archaea both can evolve by something called binary fission. And that is just basically cloning yourself and like splitting yourself into half. So E. coli are a famous model organism, meaning that they are studied a lot. And so it's good to study bacteria because they reproduce very quickly and they don't need many nutrients. And so E. coli can divide once every 20 minutes. So imagine if just like every 20 minutes a new you appeared in your life. I don't think I would be happy with that. Anyways, so yeah, again, also a model organism just means that they are used a lot to carry out experiments in science. So for example, if I want to see how this medicine will react in a human body, I can first put it in a bacteria and see how the bacteria reacts. Then I can like implement it in my own. So endospores are another adaptation of bacteria, and they're like hibernation for bacteria. Basically, everything just dies off except for a copy of the DNA and traits. So even if like the world was burning down, endospores could protect the bacteria's DNA and pass down its genes when conditions are favorable again. And so, yeah. So how biodiversity arises. Again, binary fission is a asexual reproduction process, meaning that there's only one parent, and that parent is cloning itself. However, in the process of cloning, you have to replicate your DNA so that your clone also gets a copy of your DNA. And in this process, sometimes mutations occur. Mutations are when your genes don't follow the code how they are supposed to, and they give rise to new codes. And so the high mutation rates of bacteria, like E. coli, has a mutation 1 in 10 million. That's not a lot until you realize that that's more than 2,000 mutations happening in one day. And so you have to account for the fact that mutations can happen, and that's how. Okay, wait. I don't think I fully explained this, but mutations can happen, give rise to new traits, and then if these traits are favorable, it can be passed down. But the chances of a mutation being favorable versus like it harming you, it's too great of a risk. So many organisms, and rather than relying on mutations for diversification, they perform something called genetic recombination. It's where you get your DNA from two sources. So we are technically an example of genetic recombination because, you get your genes from your mom and your dad. But this is not the same as sexual reproduction because there are no like sex organs involved. It's just like a lot of it is human manipulated. A lot of it is just up to random chance. It's very weird. Yeah. So the first kind of genetic recombination is called horizontal gene transfer, and it's exactly what it sounds like. You pass genes on through a sex pilus. No, that's a lie. Wait. Yeah, okay, that's a lie. You just pass on your genes, I guess. I'm pretty sure it's through a sex pilus. I will research this when I get off this Zoom. So yeah, disregard everything I said. Okay, the second type of genetic recombination is something called transformation. And this is when the traits, either the genotype or the phenotype, a genotype is the set of an organism's full set of traits, and then the phenotype is like its appearance. And so it can turn harmless bacteria into harmful when it's in. Wait, okay, so if you have like a disease strand of DNA and then it inserts it into the environment, it can turn a harmless environment into a harmful one. And so basically the chromosome takes in an allele and replaces it with its own. So if I had a banana hating gene, basically I hate bananas. Who here likes bananas?
Me.
Okay. Alexa, okay. So just imagine if I took my own banana hating gene out and I replaced it with all of your banana loving genes. I would become a mega banana lover. Helen, that emoji scares me. But yeah, that's how we can change the genomes of bacteria this way. And create new bacteria. Transduction is when phages carry genes from one host cell to another. So phages are viruses that attack bacteria. Conjugation is when two bacteria are connected through a sex pilus, and then it's also called a mating bridge, and it's hollow, and then the genes pass through that So horizontal gene transfer just is a general umbrella term for all of these three functions. Okay, so someone said, Vedant said, this is kind of random, but when they genetically alter the organism's DNA, it doesn't affect the organism moving so far, but affects the babies of the organism, right? I think that's kind of a like case by case scenario, because if someone just like took out like, I'm trying to think of an example. Okay, so like when you move, you have to depend on energy from like calcium, or no. Okay, I have a better example. So glucose is like sugar in the body and it's energy. And then when we don't want glucose in our bloodstream, which contributes to high blood sugar, we use something called glucagon, which is a hormone, and that builds up glucose into a stored version of glucose called glycogen. Now imagine if someone just like made it so that the genes coding glucagon just didn't exist anymore. So I wouldn't have that hormone, and I wouldn't be able to build up glucose and store energy. And so this might affect my babies, but I think ultimately it depends on like what like gene you're editing, because either way it's going to affect you because it's your genome, but sometimes it doesn't affect the babies. So it's like a case by case thing. I don't think I answered that question. I just think I went on a tangent, but yeah. Okay, so there's something called the Gram stain. And so the Gram stain is a laboratory technique that can separate most bacteria into two main groups. So the first step of the Gram stain is to get these bacteria cells on a microscope slide, and then you soak it in a violet purple colored dye, and you treat it with something called iodine, and then, or iodine, I don't know. Anyway, so then these cells are washed with alcohol, and then they're stained again with a red dye called or often, the red dye often used is called safranin. I think that's how you pronounce it. Anyway, so gram-negative bacteria. Okay, so yeah, sorry. There are two different types of bacteria, gram-negative and gram-positive. So gram-negative bacteria are identified when you wash out the dye and it is pink and red. So that means the violet dye does not stay there. And so this means that they have a very thin cell wall made out of peptidoglycan. On the other hand, gram-positive bacteria retain the blue dye or violet dye, and they have super high levels of peptidoglycan in their cell walls. So that's the main difference.
Is the purple cells alive or dead?
That's a really good question that I don't know the answer to. I want to say because they've been like died so many times that they probably aren't alive, but let me actually Google this right now. Are bacteria in gram stain alive? Yeah. Okay. So all bacteria are killed during the gram stain because of the heat and the chemicals. So they are not alive. Okay, so another major way to classify bacteria is based on shape. So we have three different kinds of shapes. We have the sphere, we have the rod, we have the corkscrew, and I think this slide is very self-explanatory, so I'm just going to let it speak for itself. Oh my God. Wait, look at these Zoom stamps. Okay, sorry, my bad. Clear all drawings. Let's move on. Okay. So actual biodiversity. Wow. So these are all of the different ways that bacteria are classified. Again, the really whack thing with bacteria and archaea is that you can't really classify them into like phylums and species because there's just so much that we don't know and we haven't explored. So we go off of things like the gram stain. So first we have low GC gram positives, and these are also called firmicutes. And so this means that they have a low GC to AT ratio of nucleotides and DNA. So DNA is made out of 4 like building blocks, and they are ATGC. So it's like DNA is like the ladder, and you have like 2 steps, and G and C are the building blocks that go perfectly. A and T are the building blocks that go perfectly. So that means compared to A and T, these bacteria have lower G and C building blocks. And so the name is misleading because some of them are gram negative, some of them don't even have a cell wall, so you can't use the gram stain. But yeah, you have all of these different kinds of bacteria found in them: Clostridium, Bacillus, which causes anthrax, Staphylococcus, which causes a lot of diseases like, I want to say like syphilis, but I have no idea. So someone asked, wait, so what do they do in the gram stains again with bacteria? Okay, so let's go back to the previous slide. So first you get these bacteria onto a microscope slide, and then you soak it with the violet dye, and then you treat it with a chemical called iodine. And then you wash it with alcohol, and then you stain it again with a red dye. And basically, if the bacteria are red, then that means they're gram negative. But if the bacteria ends up being the same purple color as the first time you dyed it, then that means they're gram positive. And just basically know that gram negative bacteria don't have a lot of peptidoglycan, while gram positive bacteria do. Okay. So high GC gram positives are also called actinobacteria, and unlike low GC positives, they have high GC to AT ratio. And so many of these bacteria include branched bacteria, and they are like fungi. Fungi reproduce by forming chains at spores. So like little mushroom heads, that would be an example of a spore. And tuberculosis is part of this actinobacteria move, and streptomyces, which are famously used for antibiotics, they are also part of this group. So a really important thing to note about bacteria is that they're not all like bad. You have all of these disease-causing bacteria, but then you also have bacteria that can create antibiotics that arise when you ferment yogurt and are really yummy. And yeah, just the sheer amount that we can use bacteria for, there's just a lot of uses, and bacteria are not all bad. Basically, I am a fan of bacteria. Anyway, hyperthermophilic bacteria are heat lovers, yeah. Examples include aquifex, which are found in volcanic vents, and thermotoga, which are found in oil reservoirs. So have you heard of PCR? It's okay if no one has. But anyway, PCR, okay, so up until recently, like sequencing our DNA sequence has been a really hot topic. So as Vedant said, PCR, or polymerase chain reaction, is a way where we can clone DNA like super fast. So basically PCR just turns one copy of DNA into a whole bunch really fast. But this PCR cannot be possible without Taq polymerase, which is a kind of bacteria that can help DNA replicate itself. And so Tachylomeres belongs to Hadobacteria. Cyanobacteria are blue-green bacteria, and they're the first photosynthetic organisms on Earth. So people believe that they are the reason plants have chloroplasts and can photosynthesize. In the interest of time, okay, sporochites are gram-negative bacteria with axial filaments, so they're like a corkscrew. They cause Lyme disease, syphilis, as you can see on the right. That's a spirochite. Biodiversity part 3. I don't know why I read that slide name. Anyway, chlamydias are the smallest bacteria. They are parasites, gram-negative. Proteobacteria. They are the largest group of bacteria, and they basically include everything. And just like how people thought that chloroplasts came from cyanobacteria, people think that mitochondria came from proteobacteria. So that's pretty cool. Anyways, blue kit time.